Hello, everybody, and welcome back to day 3 of your Earth and Environmental Science course. So today we're going to do things a little bit differently. You're going to watch this recorded lesson that goes over the scientific method. And this is going to be a little bit easier for you because you do have your guided notes that you should have found in your Canvas course. So as you watch this, you can pause and you will delete the blanks where the blanks are in your notes and you will fill them in with the information in the presentation. So what's really great about this is that you can pause it whenever you need to fill in your notes, and then you will keep those notes in your Google Drive folder along with all of the other notes that we'll do during the semester. So we're going to get started today learning about the scientific method, which is going to be an important concept that really comes up in every single one of your science classes that you think of. So your warm-up here is I just want you to take a second and think of what do you think of when you hear the word scientist? What does that word mean to you? And for you guys, I know myself when I was younger, when I thought of a scientist, the first thing that popped into my head was always somebody in a white lab coat working in a laboratory somewhere with pipettes and maybe blood samples, maybe CSI, like crime scene investigation. That's like what we think of typically when we think of a scientist. But what would you think if I told you that you are actually a scientist? You would probably think, what? What do you mean I'm a scientist? Last time I checked, I was a student at Rocky River High School. I'm not a scientist. Well, it's true. We are all actually scientists walking around in everyday life because we use something called the scientific method. Now, the scientific method is a proper term that we use to describe kind of the process that scientists use to draw conclusions about an experiment that they're running or a question that they have. But what we can find is that we also use the scientific method in everyday life and we don't even realize it. So what is the scientific method exactly? The scientific method is an organized way to solve a problem through experimentation and observation. Okay? So when we say experimentation, we often mean in a contextual setting of a science lab, actually running an experiment, recording your observations, and then drawing conclusions about what it is that you're seeing. Okay? So the scientific method is just the steps that you're going to take to get to that conclusion. So there are 6 steps of the scientific method that we're going to learn about today. Step 1, we are going to state the problem. So if you are in a classroom and you're starting an experiment or you've made an observation and you have a problem, you have to actually figure out what the problem is before you can get started. Okay? And then you are going to form a question about a specific event. So we're going to use an example today that I'm going to take you through to kind of explain each part of the scientific method. And the example today is mouthwash kills bacteria. Which brand works best? Okay, so this is an example of kind of everyday life where you're out and about and you have a question, you make an observation, and then you have a question about that thing. So we know that mouthwash is marketed to kill bacteria in your mouth. That's the point of it, right? But you want to know which brand works the best, right? So that is the question that you're going to present. And we're going to use the scientific method to come up with an answer. Okay? So our second step in the scientific method is going to be collect background information. So we are going to research the problem. And when we say researching the problem doesn't have to be accessing academic journals and finding, all of this complicated information. Researching the problem can be as simple as going to Google and typing it into the search engine and seeing what comes up. In this case, when we have mouthwash, where we're going to see which mouthwash works best, research would literally just be taking those mouthwash bottles off the shelf and then reading the ingredient list. So an example for this, you're reading the labels of the types of mouthwash to see which one has the most antiseptic. And an antiseptic is a germ-killing ingredient. So you would make or have the question of which mouthwash kills more bacteria. And then to do your research, you would look at the percentage of the antiseptic that is contained in the mouthwash. So it doesn't have to be anything too intensive, but we do have to research. Step 3, you are going to form your hypothesis. Now, a hypothesis is very important. You will see this word over and over in your science education. And this is a testable, educated prediction based on your research. So you are predicting, based on what you already know, what the outcome of your experiment will be. It can always be recognized as an if-then statement. So if we are going to state a hypothesis, we will say, if this happens, then this will be the result. And what do I mean by that? So in this particular example, we say, if Listerine has more antiseptic ingredients than Crest or the Walmart brand, then it will kill more bacteria. Okay, so you can always spot a hypothesis by observing the if-then statement. And if I ask you to give me a hypothesis, you're going to want to write it this way in particular. Step 4, we're going to design our experiment. So you have to decide and set up the actual experiment that you're going to run to test your hypothesis. The whole point of an experiment is to test your hypothesis, because remember, your hypothesis is a prediction. You don't know if it's right or not until you actually do your experiment. So you're going to create a controlled experiment, okay? So when we design an experiment and we say it's controlled, it is very important that we control certain parameters of the experiment to make sure that our results are valid. So a controlled experiment must have 2 groups, okay? So this is what we mean when we say controlled experiment. The control group, where all conditions remain the same. So it's a group that you don't do anything to. Okay, so it's a group in the natural state. Control groups are important because without a control group, your results mean nothing. Okay, let me say that again. Without a control group, your results of your experiment mean nothing because the control group serves the purpose of a comparison. You compare your results of your experimental group to your control group to see if there's been any change. You can't tell if there's been a change in the actual process of your experimentation without a control group. So a control group is very important. And then the experimental group, on the other hand, is the group or the many groups that you test. So the experiment is performed on this group. Okay, so control, all conditions remain the same, and then the experimental group gets the actual experiment performed on them. So you have to establish your variables in your experiment. The first is the independent variable, and this is the variable that you change or manipulate. And if you were going to graph your results, this would be on your x-axis, and your x-axis is the horizontal. Okay, so if you're talking about a graph, that's your horizontal. So the straight across. And the way that I always remember this is that the independent variable is the thing that I change. It's the thing that you manipulate about the experiment. And the dependent variable is the change or the result that happens when the independent variable is used. And when you are graphing the dependent variable, you are going to graph it on the y-axis. Okay, so the up and down axis. So the dependent variable, the way that I always remember the dependent variable is that it's the thing that you measure. It's the thing that you're changing. So in this experiment, if we're measuring how much bacteria is killed by the mouthwash, the dependent variable is the amount of bacteria that are dead, because that's what you measure in the actual experiment. So here's an example of how we would set this up. So we would have our control group at the top. So these would be 4 petri dishes where we would grow bacteria, and then we would test these mouthwashes on these bacterial cultures. So on top, we have our control group that has no mouthwash. All right, remember our control group is important because it doesn't get the experimentation, right? This is what the bacteria would be doing on their own in their natural habitat. They're living life, they're loving it, they're not coming into contact with any form of antiseptic. And it's important because we have to compare our experimental results to the control. Without the control, our results mean nothing. And then our experimental groups are going to be the 3 Petri dishes that have the Listerine, the Crest brand, and then the Walmart brand. Okay, so here our independent variable is the thing that's changing, the thing that I change about the experiment. In this particular experiment, the thing that we are varying, the thing that we're changing, is the mouthwash, okay, the type of mouthwash. We've got 3 different types. We're varying the types. That's what I'm changing. The dependent variable, again, is what changes as a result of the change in the independent variable. So it's what we measure. So the dependent variable here is going to be the amount of bacteria killed. All right, do we see how this works? The dependent variable is the amount of bacteria killed. So the amount of bacteria that are killed depends on the type of mouthwash that's been applied to the bacteria. Okay, the dependent depends on the independent, and the independent is what we manipulate. Step 5 of your scientific method is to collect and analyze data. You're going to run the experiment, so do the actual experiment, and then you're going to write it down. There are 2 types of data. Quantitative data is numerical data, so this deals specifically with quantities, usually in the form of a graph or a chart. So an example of quantitative data would be there are 5 desks in the classroom. The gummy worm is 3 inches long. Okay, if there is any kind of numbers involved, it's quantitative data. Qualitative data is descriptive data. It describes how something looks, smells, sounds, or feels, and it deals with quality. So an example of this would be the room feels cold. It smells like roses. Okay, so quantitative, there's a number involved. Qualitative, it describes the quality of something. So here's an example, a qualitative example. The control group and the Walmart brand have a strong smell. The Listerine and the Scope don't smell as bad. We're describing the smell, so that's qualitative. It describes the quality of the smell. And then the quantitative example, this example here is called an area of inhibition. That just basically means the area where bacteria is not growing on the Petri dish. So if we look, we've got our control, our Listerine, our Scope, and our Walmart brand. The larger the area, so the larger the number in this chart, that means the larger the area where bacteria cannot grow because the mouthwash is doing its job and killing the bacteria. So if we look over time, if we look at our control versus our Listerine versus our Scope versus our Walmart brand, our Listerine ultimately has a largest area of inhibition, meaning that it's killing the most bacteria. And so then step 6 is to draw conclusions. Determine if the data collected actually supports your hypothesis. So was your hypothesis correct or incorrect? So based on our quantitative data, the Listerine did kill more bacteria than other brands because it had more antiseptic ingredients. Okay, so we're going to accept our hypothesis and we're going to say that yes, Listerine is the best type of mouthwash. Okay, so that is your scientific method. Today we're going to do some classwork and some practice with that. Make sure that you have your notes completely filled in, and then we will come back together and do some group practice with this. If you have any questions, let me know.